Can you hear me now? Okay, perfect. Hello, everyone. Thank you, Diego. This is my presentation on FinaleToolkit. So first, to understand FinaleToolkit, let's go over what exactly is cell-free DNA. So cell-free DNA are these highly fragmented pieces of DNA found in a variety of body fluids, including blood plasma and urine. And it's been shown to be a rather potent biomarker for a variety of pathological and physiological conditions, including cancers, organ rejection, and for prenatal screening. And one particularly useful part of cfDNA is that the fragmentation pattern contains a lot of epigenetic information from the tissue or cells of origin. So here's just an example. A lot of cfDNA in healthy people is created through apoptosis. So imagine a cell that is going through this process, right? So nucleases will start to digest the DNA within the cell, but this process is not even, and it can be affected by a variety of these epigenetic factors, including, in this example, we see that the DNA surrounding these nucleosomes, these histone octamers, tends to remain behind more often than the linker DNA between, which is more affected by the nucleases. So this is just one example of how epigenetic information is stored in the fragmentation pattern. So numerous cfDNA fragmentomics features have been characterized, which extract epigenetic information from whole genome sequencing of cfDNA. And so here's an example of the window protection score described in a Cell paper in 2016, which looks at these WGS sequences of cfDNA and extracts the nucleosome spacing from them by using a rather clever scoring system. So yeah, this can be useful for determining tissue of origin, for example. And so other features have been described, like 5' M motifs, motif diversity score, Delphi, and much more. However, we run into a problem in terms of software availability. So sometimes authors don't include the code that they use in their software. Other times, it might not be runnable in your specific context, or it might not be optimized for really large fragmentation data sets. So that's where FinaleToolkit comes in. FinaleToolkit is a Python library and standalone software toolkit that generates these fragmentomic features based off of sequence alignment maps. So as you can see, the features I mentioned before. FinaleToolkit can generate these pretty accurately and precisely. So in this example, you'll see a comparison between the window protection score generated with FinaleToolkit and from the scripts provided by original authors, and the correlation is pretty good. Here is an example where we did not have access to the author's data. So you see here N motifs and motif diversity score on the left and right respectively, versus the data we generated trying to replicate the results. And it's also pretty close. FinaleToolkit is optimized in a few ways. So this is an example of how the runtimes improve, in this case by 5 times. But FinaleToolkit is also parallelized, so we can see up to around like a 20 times improvement. 50 times improvement in the runtime speed with 16 cores. And this applies to all the other features too. So, and in some really extreme examples, we notice a really great increase in the memory efficiency of our toolkit. It's well documented and easy to use. You can download it now into your computing cluster or even your personal computer using pip. And there's also online documentation, both for the command line interface and for the Python API. So in summary, it's FinaleToolkit can comprehensively generate accurate cfDNA fragmentomic features. It's fast, parallelized, and memory efficient, and it is user-friendly and well documented. I'm open for questions.